Let's start back again. Our next speaker now is gonna be Kevin Vance. He will give you also a talk on Bio-Deuteration. Kevin, do you want to share our screen?
Are you able to see my screen now?
Yes.
Looks like there's some people still joining us. I'll go ahead and get started. As mentioned earlier, my name is Kevin Weiss. I'm a member of the Biological Labeling and Scattering Group here in the Large Scale Structure section and the Neutron Scattering Division at ORNL. And my focus here is really to talk about biodeuration. If I can advance my slide. So the Bio-Deuteration Laboratory is the central facility here and user program for deuterium labeling of macromolecules. And it's really to help support the neutron structural biology program here at ORNL across multiple beamlines. But in today's focus will be basically on deuteration for sands. And Hugh mentioned some of the other applications earlier in his talk. And what we're trying to do is develop efficient methods to produce these materials. And one of the main components is that we train students, users, and staff in the application of these techniques. So maybe you don't produce large quantities of protein in your own lab because of the type of work that you do or the complementary techniques that you use. We have the facility and ability to train you in applying these techniques for your neutron experiment. And I also we also assist in approving the downstream efforts. An example that Luke talked about earlier is I've been heavily involved in the SEC SANS development project. So as was mentioned earlier, neutrons are an excellent non-destructive probe that can discriminate between hydrogen and deuterium. And for small neutron scattering, it's great for characterizing higher order macromolecular complexes where we partially deuterate one of the components and get structural information regarding each individual component, all in the same complex, for example, a protein complex. Other techniques like neutron protein crystallography utilize deuterium to allow them to pinpoint positions of individual hydrogen atoms. And the deuteration there is a fully deuterated molecule where they're trying to maximize the coherent scattering signal and decrease the incoherent background. And then, as Hugh mentioned, we can also probe structure and dynamics about biomacromolecules, and some other labeling techniques can be used for that as well, mainly segmental labeling or residue selective labeling, so that you can look at individual domains or dynamics of individual residues against a background of a per-deuterated background where you're just looking at the hydrogen signals. So what is biodeuteration? And for the purposes of today, this talk, it's really the production of biomolecules in a D2O media. So we're using organisms, growing those organisms in D2O media, sometimes with a deuterated carbon source to deuterate the biomolecules. And we're specifically deuterating the non-exchangeable positions. Of course, the exchangeables are deuterated as well, but those come and go based on the percentage of D2O that you're dissolved in, whether you're dissolved in water or D2O. But in general, as you increase the D2O concentration in your media, your deuterium incorporation in your biomolecules will go up. And this plot shows that. And once you add a deuterated carbon source, you can produce in a high D2O concentration media, you can produce what's known as a per-deuterated biomolecule. And early experiments in this involved folks at Argonne National Lab, Katz and Crespi, where they're growing green algae and cyanobacteria in D2O using CO2 as their carbon source to study the effects of D2O on organisms. And what's that has turned into is a valuable source for fully deuterated materials. And those extracts have been used and actually are sold to culture other cell types, or even some cell types that do grow in D2O as a supplement for producing biomolecules that are deuterated. But in the 1970s, the advent of recombinant DNA technology has allowed us to do more in terms of deuteration, where we acquire a gene of interest, prepare an expression vector, transform our cell type, and then grow those cells, induce expression, and then purify the protein. So when it comes to deuterated protein expression purification, it's really the same process. One step has been added here where we adapt the cells to D2O media so that they can grow well at a small scale before we scale up and grow them and induce expression. So as a training or a workshop, I want to give a chance to kind of go through the process, and please put questions in the chat or raise your hand if you have any questions, and I'm glad to stop and take those questions. But the main recommendation that I have for people starting this type of work is you probably should optimize your unlabeled production before you even think about doing deuteration. And that's by selecting the right construct or vector that works well, the strain that works well. You might vary this concentration of your inducing agent to get better protein production. It might require temperature adjustments to maximize soluble protein expression. You might even have to do more work on your extraction and purification method to determine what's really the best way to get most of the protein out of the organism once you grow and express the protein. Other things could be done by doing a flask-based experiment or a bioreactor-based experiment. Sometimes it's worthwhile if you visit to do deuteration here. Sometimes it'd be worthwhile to do a bioreactor experiment on an unlabeled test to try to confirm what we can get out of using a bioreactor scale prep, because those are different experiments. One thing we can do in advance of a visit here is help you work with minimal media. We can send you the recipe that we use here in the Bio-Deuteration laboratory for our bioreactor experiments. We can send you the deuterated version of that, and you can do some small scale tests in your home lab, run a gel, determine that you can actually produce the material of interest in a minimal media before committing to come to do a deuteration experiment here. And what do I mean by minimal media? If you're not familiar with it, it's a media that has no rich media extracts, no amino acids or precursors. It's just the basic building blocks needed for the organism to grow. In this case, E. coli media, where you have these salts and add a carbon source like glycerol or glucose, but no other components. And that allows us to tune the D2O content and tune the deuteration content of the macromolecule that's produced. For a per-deuterated media, that also has no source of hydrogen. Sometimes we would even exchange the hydrogens on the salts ahead of time in a rotary evaporator. Also would not have any amino acids or precursors. So you can really control everything about the media, removing all source of water, and then you produce, using one of these deuterated carbon source, can produce a molecule that's fully deuterated, meaning per deuterated denotes that the maximum proportion of deuterium has been added to that molecule. You also can add deuterated amino acids as a carbon source, but that is at a cost of the financial cost of making a more expensive media. So some tips for working with D2O and deuterium media in general: you're going to use dry and sterile glassware, and we sterilize that media by sterile filtration, so no autoclave. We autoclave all the glassware ahead of time, dry it in an oven. The media itself, we're trying to minimize prolonged exposure to air. And for the bioreactor, we're actually blowing quite a bit of air, several liters per minute through the vessel, and we dry that air using a gas purifier, a dryer, to maintain a dry air circulation through the system. And this last box at the bottom just shows. HR is hosting a lemonade stand. Sorry, we have an announcement here. So this box on the bottom shows what happens if you're to leave like a petri dish size dish of D2O out on the bench. It'll exchange, half of it will exchange to H2O in about a 2-day period. So that's kind of an extreme. We wouldn't necessarily have this sort of setup in the lab, but it just shows you it does exchange with the normal H2O vapor in the air, and that we should do our best to maintain kind of either a sealed container or dry the air or some way to protect this from happening. So a little bit about D2O adaptation. There's a couple of different ways you can go about this. One method is where you prepare your rich media plate and then just slowly increase your concentration of D2O in your minimal media up to the percentage of D2O that you would like to use, which would then convey the level of deuteration that you're looking for. So that's kind of a time-intensive and kind of babysitting type step that has to happen. Another way that you can do this is do solid media transfers, which is a little bit easier in terms of the process itself. You can just transfer between rich media to your H2O minimal media plate, and then go to a D2O minimal media plate. And that D2O minimal media plate has obviously been autoclaved to be able to melt the agar, so it's not highly deuterated because of the exchange that's happened. But at the very least, you produce a D2O minimal media plate that once those colonies start growing, the D2O content of that plate is high enough that you can transfer individual colonies straight to high level of D2O minimal media. So back to the process, we can actually avoid that adaptation step altogether. And that's by using these extracts that I mentioned earlier. There are solutions commercially available and powders commercially available that are listed here. I won't list them individually myself, but those are commercially available, a little expensive, but those can be used as minimal media additives and even whole solutions that basically mimic a rich media or LB media that allows you to produce these deuterated cell pastes without an adaptation step. But there is a cost to that. So trying to pause a little bit. If there's any questions, I'd be happy to take any so far. So some practical tips and suggestions when you first start these type of experiments.
Kevin,
Yes.
sorry, you have a couple questions.
Sure. I guess I can't see when I'm presenting I don't see the hands raised.
Hi. I have a question.
Sure.
Um, uh, if the plant is growing in D2O media, so how you are exactly control the water vapor when the plant is growing in D2O media?
So you can grow some plants in D2O. When I'm talking about what I'm kind of focusing on in this presentation, because most of the deuteration that happens involves protein expression in E. coli or yeast. Those are suspension cultures where you're shaking them in a flask or growing them in a bioreactor. For plants, people have developed ways of doing that. Barbara Evans, who works here at ORNL, has done a lot of work on that, where she's growing the plants. I believe it's switchgrass has been grown in
Yes, so
a,
Kevin, I think
yes,
I can help you here. So
sure.
so if that is going to be a plant, it's usually grown in a closed environment. So you can add the amount of salt, like amount of salt, add into the D2O medium, control the amount of the humidity generated inside the chamber. So there are techniques that you can control the humidity control in the closed
And
environment.
yeah, and she's using these tall graduated cylinders where she controls and kind of keeps that sealed and varies the D2O content in the the growth media to allow her to control the D2O content in the resulting product. Now, I will say there's a certain concentration of D2O. Once you get to that point, it may not grow anymore. And so there's limitations to that. You can't, and this is true for all organisms. Not all organisms grow in D2O, and some of them will kind of tap out and not grow any longer once you reach a certain percentage. Now, things like E. coli and yeast um, are the two main ones that have been used quite often to produce these materials, and those can be grown in 100% D2O.
Okay, thank you.
No problem. Was there another question?
There is one in the chat, Kevin. It's from Thomas. Um, he said that D4 acetic acid is $118 for 50 grams, but D7 glucose, it's 300 to 500 per gram. So I'm assuming that acetate has the carbon source is preferred.
Yes, so I didn't touch on that. I'll go back to that slide. So as you go from glucose down to sodium acetate in that list there, the cost does go down as you go down the list. But there is another cost to that is the growth rate and yield of final product. And what we've settled on for per deuteration is to use a deuterated glycerol, kind of compromise between those. Now, for the purposes of a SANS experiment, you don't always need a fully deuterated per deuterated media. So oftentimes we're not even using a deuterated carbon source, but there are experiments where you might want that signal, where you would want a high percentage deuteration, and that's where you would add the carbon source. Was there any more, Wellington?
No, you can move.
Okay. So some practical tips before you start. These are long growth times. So my recommendation would be work from a fresh transformation. We ask people to avoid using ampicillin resistance. The beta-lactamase that's produced in these systems does leak into the media. It can chew up the ampicillin there, and then you end up at these long growth times, you can end up growing cells that don't actually produce the protein because they don't have your plasmid of interest in there anymore. It's not mandated. We do these growths with carbenicillin as a antibiotic and have been successful with that. But it's just something to think about if you're starting, from the beginning. We generally like to use kanamycin as a resistance marker. And if you are using a beta-lactam antibiotic like ampicillin or carbenicillin, we tend to centrifuge resuspend the cells before we inoculate the next step in terms of scaling up for protein production or even in the adaptation process. And one thing to note about these D2O growths, as you go above 50% D2O, upwards of 75% D2O, you have to use more inoculum to prevent these really long lag times. So whereas maybe normally you're doing more of like a 1 in 100 or even more dilute inoculation as you go from one culture to the next. You might need to do a 1 in 10 or a 1 in 20 dilution in order to maintain the culture. And that's kind of system dependent in terms of the type of cell that you're using, the strain that you're using. But in general, 1 to 20, 1 in 50 in that range is something a little more concentrated than what you're maybe normally used to, where you can just pick a colony, touch it to your media, and then have something grow well. And then the other point about adaptation is the main adaptation is to D2O. We find that we can adapt to D2O and then add the deuterated carbon source later on, kind of to preserve the use of like an expensive carbon source until we're about ready to scale up. So we adapt fully to D2O and then add the deuterated carbon source. Last one is to do these small scale tests in a H minimal media and then a deuterated minimal media before you commit to doing the full batch protein expression in a large shake flask or even the bioreactor. And as I talked about a couple different times, the challenge is the scale up and the cost. For these neutron experiments, we're doing multi-milligram scale preps, and oftentimes you want a range of different contrast points where you're dissolving your final dialyzing or dissolving or whatever, you're producing your final solutions for your SANS experiment in multiple percentages of D2O, and that can add up over time in terms of the amount of material that you need. Already mentioned the fact that the growth rates reduce and you get less biomass out of these minimal media growths and often less yield in terms of the protein produced. And mainly that's most likely due to the fact that you have these normal enzymes that normally use water as part of the reactions, and they're used to that water environment, and you have the isotope effect going on where it can change basically the biology of the cell based on the enzymatic reactions that are going on. Things slow down. The other challenge is the fact that we're very limited in the types of media that we can use because we want to control the D2O. We want to control the D2O content and the deuterium content of the final product, and you can't do that if you're not controlling your media. So that limits what you can use. And then, of course, as I mentioned earlier with the plant question, there are host limitations. Some organisms don't grow well in D2O. Thankfully, we have some cell types that do grow well and that we can do high level deuteration with. Now, I'm going to go into some examples. This first example is an example of a per deuterated production, but this can also be applied to partial deuteration. So in this case, our expression host was BL21DE3. We were using a codon optimized gene, which had kanamycin resistance. We used the solid adaptation process where we grew the cells on the D2O agar and then transferred to 100% D2O media prior to scale up. We were using a bioreactor in this case, so we have a batch amount of carbon source in the bioreactor, and then we produce a 10% feed solution that we feed then on top of that initial 5 grams during the process. And then we coordinated induction with the initiation of the feed and then harvested after induction using IPTG was used for induction. And what do we get from using a bioreactor? We get obviously higher cell densities than we can in a shake flask because we have better aeration, better control of variables like pH, the temperature, able to mix because there's a stirring motor attached to the top. We're able to, we actually just added the capability to measure the backscattering to get an OD measurement at the vessel So we're going to be testing that soon to add as another variable that we can use in the process. And then at the side, we have the peristaltic pumps that allow us to feed in the carbon source, and we use the dissolved oxygen signal to trigger that feed. So this is what our minimal medium for fermentation looks like. Looks basically the same as the minimal media that I showed you earlier with a couple of differences. Main difference is we have the magnesium there and trace metals, and an extra source of or increased amount of ammonium sulfate for producing in the bioreactor. And this base media that we've used for many years allows us to get cell densities upwards of 25 OD, whereas in shake flask, you might not be able to reach that high with a regular M9 minimal media. So that's another picture of the bioreactor, and I'll go by this in the virtual tour a little bit later. It won't be assembled in the tour. So this next figure shows what happened during that process. As you start the experiment, your dissolved oxygen trace starts dropping as the cells grow, become more dense. They're consuming more of the dissolved oxygen, so your levels drop. Once you reach a certain point, we set a set point of 30% dissolved oxygen. That triggers the agitation to start. So that's the red plot where the stirring ramps up. And then at some point, that initial carbon source is exhausted. The D2O or the dissolved oxygen spike happens. That triggers the feeding from the pump. So that's the third graph there that shows the 10% glycerol being fed in throughout the course of the end of the experiment. And we also control the pH or PD by adding an NaOD solution. Once the PD drops down to a certain point, it triggers the base pump as well. So one question we often get asked is, how do you afford all the D2O for the protein purification? Well, we don't do that. Since we're only worried about the non-exchangeables, they're not going to exchange back during the protein purification. So when we lyse the cells, spin them, and do our chromatography steps, that's all done in H2O. There might be an exception where if you had a heat treatment step as part of your purification or your lysis process, that heating step in the process to try to precipitate proteins that you don't care about might actually have some exchange happening. So there you might want to think about doing that in D2O. But for the most part, the protein purification is always done in H2O. And just some results from that particular run, just to show you that this is kind of our all-time gold standard of experiments. So it's probably a bad example to show everybody, but from about 16 grams of wet cell paste, we were able to get 600 milligrams of per-deuterated beta-lactamase for that experiment, which happened to be for neutron protein crystallography. But for SANS experiments, the process is basically the same. We can use the bioreactor to do this. We just use a different percentage of D2O. So once you have a protein purified, this particular example showed that I'm showing, they concentrated the fractions exchanged in D2O buffer and then crystallized in a D2O condition, produced really large crystals for neutron protein crystallography. So that's one way of producing proteins. Another way that I'll mention is this idea that you might have a system that just doesn't behave well in minimal media, period, either H2O minimal media or even D2O minimal media. One way around that we've found was useful, it's been used by the NMR folks for years and years, is you grow your organism of interest in H2O media, grow up your large pellet in a rich media, then pellet them down and wash with D2O media, and then resuspend it in that media and induce. And in this particular example, we're inducing at a 4x concentrated media, so that conserves some D2O. And we've done this with different percentages of D2O media successfully. And as you get towards 85% D2O medium, you can produce proteins that have a protein match point of around 100% D2O for your SANS experiment. Sometimes that's useful in the sense that you were trying to hit 100% D2O match point, but a lot of times we're reduced down to 75%, 80% range so that we can actually match out at lower percentages of D2O, something that gives us enough contrast from, let's say you're doing a protein complex where you want to label one component and mask that one, and then flip it and mask the other component. You need to be able to match both of those out. So that's in those types of experiments, we're actually producing proteins in media that's lower than 85% D2O. Otherwise, you won't be able to match out one of the two components.
Hi, Kevin.
Yes.
We are 5 minutes from our next break.
Okay.
Okay.
Break or the tour?
Break. We're gonna give the tour after lunch.
Okay.
Okay.
So contrast variation experiment, as Volker mentioned, I won't spend a lot of time on this. There's natural contrast in biomolecules, so you don't always have to do labeling unless you need that contrast between either two individual proteins, or maybe you need more signal to separate your protein from a nucleic acid, for example. And then you vary your D2O buffer to get information about individual components in the complex. And by selectively deuterating an individual protein, we can look at a protein-protein complex by varying the D2O buffer in our SANS solution. So one example of that shows for SANS, we did some experiments during the COVID outage where we were producing deuterated papain-like protease and binding that to ISG15 and looking at the conformational landscape of the ISG15 in the context of the protein complex. So it was a combination of SACS and SANS experiments where we were able to partially deuterate the PL-Pro and then match it out and look at what's happening to the unlabeled ISG15 in the SANS experiment. So a couple of comments on how to get access to the Bio-Deuteration laboratory. Our access model is if you have an approved neutron experiment at one of the Orano beamlines, like BioSANS, you can get access to the Deuteration laboratory. So we have a information request form that we send out to folks, have you fill out your information, who might be coming to participate on site in the Deuteration experiment, fill out all the information that you have on your current system, helps us start the dialogue about, how we might start a project, and gives us a baseline of where we are, and then we can work with you, send you some media, do some testing in your home lab before you commit to a large scale prep. And those materials are paid for when you come. So you basically come, you're going to pay for your travel and lodging, but we'll support everything else while you're here in terms of your training and the materials needed. And when you fill out a proposal for your neutron beam time, you select your neutron instrument first, but as a second instrument in your proposal, you can select the deuteration lab, and that will couple your neutron experiment with the deuteration. And what we end up doing is screening experiments for feasibility for the deuteration, and that's what that information request form helps us do, is to determine whether we can really accomplish what we want to, which is produce enough material for your SANS experiment. So with that, I want to acknowledge BER, who sponsors or funds the Deuteration Laboratory and the BioSANS instrument. but also some of the other examples I presented. These are doing, or this work is done in laboratories here at SNS, and those labs are actually supported by DOE Office of Science User Facility funding, and some work in our labs also are supported by the lab-directed research and development programs. And then you can use these references later. But are there any questions?
Okay, I don't see any questions so far. Yeah, there you are. Please go ahead. You are muted.
Sorry. Yeah. So I was asking if, let's say, we have a 2-protein complex system and we want to do a contrast variation, and let's say if one of the protein is more continuous and globular, but the other one is more discrete. So does it matter like which one we should deuterate?
Wellington, you want to take that one?
So one is more disorder.
Yeah. So one is like more continuous, but the other one is like discrete and like more scattered, like having different subunits and not continuous. So does it matter like which one we should like deuterate in terms of like scattering?
So in general, it depends how you want to plan your experiment and what you want, which one of them you are more interested. In general, you try. So there is a few points about the deuteration. What is going to be easy for you to deuterate to? What's going to be more, what to give you more yield during the deuteration? But you should target the more globular one. Which is in the case if you want to, you're gonna have high signal when you are masking the lower contrast one, which is the disorder one.
Okay.
That is one option. But also always depend how you are planning or experimenting what you are targeting to student.
Thank you.
I think everyone's going to have an opinion on this one by the looks of things. So Volker, you go ahead.
I think you said in the end it depends on what you're looking for.
I think your mic might be down, Volker. I don't know.
Okay.
Yeah.
Should probably move it here. Is that better?
Yeah. There you go.
Okay. Okay. Depends on what you're targeting. So if I understood your question right, you have one subunit that's more globular, one piece, and the other one is maybe more scattered around or disordered and stuff like that. So it will be easier to observe the globular one, no matter which way. So generally speaking, right, that will be easier to detect in the scattering. But it's the question also what you're interested in, and then there's also which one can you deuterate more easily. There is one thing that well, there's different ways to look at this, but one other way to look at this is if you can make one of them deuterated such that it matches in 100% D2O, like Kevin earlier explained, but you make it invisible there, then you see the other one really well. So for example, if you wanted to see the globular one, you could actually deuterate the other one and then do the experiment in 100% D2O, then your background will be really low and you will be eliminating the scattering from the disordered one because it matches the buffer, and then you see only the globular piece. That would probably be the easiest experiment from a small angle scattering point of view.
Yeah, we tried that, but it seems like we always have like a nice thing with the globular one, and we are having problem with the more discrete one. So it's like the data is always noisy with the discrete one.
Sure. Yeah, that's expected. So if you want to see the other one, the disordered one, you could deuterate the other one.
Yeah. Can you deuterate the globular? That's the question. If you can, that would be the right way to go. You want to minimize noise, maximize signal. So you want a solvent that has very little hydrogen, as well as increase the signal from your sample. So deuterate the globular and put it at a high D2O solvent, then you're resolving your flexible domain far better than the vice versa.
And I did not talk about this in my introduction. In fact, we haven't talked about this at all yet. There's some part of the scattering contribution that's called incoherent background. That gives you background, and then there's noise that comes with the background, and that is the highest in H2O and much lower in D2O. So apart from the actual contrast, getting away from so not matching your surroundings, there's this additional background question as well. So it's somewhat complex. We all understand this very well, but it depends on very much on the detailed question. So we should basically discuss each case specifically. To prepare for that. It's rather than trying to give a global answer that fits everybody, I think.
Yeah, the molecular mass of each subunit is very important too. So if you have a very small,
yeah, that's a good point. What is the molecular mass of the two components?
Okay, thank you.
No, what is your molecular weight of the two components?
So it's like a 800 kilodalton protein. It's a big protein.
Okay.
Yeah. So it's actually like the globular one is having like the 8 copies. It's like 500 kilodalton, and the rest is the other one, which is having 4 copies and discrete and like not continuous.
And it's about 300 kilodalton. Okay.
Yeah.
Okay, but that's good. That's a good case. So that's a large one. So it should scatter really well, either way. So.
But it's smaller pieces spread out, or are they discontinuous? They're not. It's not a continuous smaller part, right? They're small pieces.
But yeah, we are not sure about it, but we know that it's more like scattered and like not continuous. Like it's not like a 4 complex system, or like it's not a tetramer. They are like monomers which are like spread it around.
Sounds like an interesting and feasible problem to me. But you're saying that you had difficulties with that before. So I'd suggest we take this offline and have a follow-up discussion on this.
Okay. Yeah. Thank you.
There are any more questions? Okay. We should have one more people there. Let me see who it is. Didi?
She just
I think you're
answered.
muted.
I think she still has her hand up.
Yeah. But she's muted.
Oh, sorry, by mistake. My mistake.
Okay, I don't see any other questions. I think we can go for our lunch, break for lunch now. And we'll try to be back here 1 p.m. to start again. All right.
Wellington?
Yes.
How are you planning to adjust the schedule since we lost a bit of time? I guess the tour, we had to move it.
Yes, I think we can do a short tour on the beginning of my section, and then try adjust the time across the sections that we have in the afternoon.
Okay.
So, but Wellington, both our sessions are going to start simultaneously at 1, is it?
Yes.
Yes.
You have them in parallel.
So both of us needs to start a bit late.
I think so.
What's the parallel?
So too.
Okay. Because there's
no, parallel.
I'm sorry?
So the sections at 1 is not, it's my talk first, and then Word talk is later.
The parallels are only tomorrow.
Only the hands-on are separated, is it?
Yes, it's tomorrow.
Sorry. That's tomorrow.
Yeah.
You're right. My bad.
All right. We are back at 1.